Hi everybody, you won't be seeing me today, just the screen. I'm going to be talking about amino acid catabolism, and I'll be reviewing the breakdown of select amino acids from your chapter. It's not comprehensive, but I've picked some that I decided that I want to talk about. When we talk about amino acid catabolism, we're really talking about one of two things, either transamination or deamination. In transamination, it literally is the transfer of one amino group from one amino acid to the alpha keto acid, to an alpha keto acid, and aminotransferases catalyze that. Deamination, on the other hand, is just the removal of the amino group, no transfer occurs, and it's a done deal. I want to talk about the sources of ammonia in the body. And that's important because ammonia, NH3, is a byproduct of protein metabolism, and it's toxic. So the body needs to break that down. It's formed from deamination, which we just talked about. The deamination of the amide groups from glutamine and asparagine. We ingest it in some foods, cheeses and processed meats, and then lysis of bacteria in the gut. Then it's subsequently absorbed into the body. The urea cycle is the means by which we essentially get rid of ammonia, and it has to be broken down. Now, I've listed the steps for you, the abbreviated steps I might add, and if you counsel a client or a patient on the urea cycle, they're probably going to either get up and leave your office or kick you out of their room because they don't understand all the biochemistry behind the urea cycle. But what you need to be able to take away are the basic concepts so that you in turn can understand what's going on with some patients that you might be counseling or that might be clients of yours. We see toxic levels of ammonia which build up when the urea cycle isn't working correctly in patients with hepatic encephalopathy. So hepatic, meaning liver, encephalopathy refers to the brain, and they're confused. They have end-stage liver failure. Their liver is essentially gone, and their ammonia levels build up to toxic levels. And the urea cycle occurs in the liver, which makes sense if you have end-stage alcoholic liver disease or cirrhosis. The liver isn't working the way it should or properly. So I've listed the steps for you. Essentially, we have ammonia combining with carbon dioxide or bicarbonate to form carbamyl phosphate. And then there's a reaction that occurs with ornithine, and you get citrulline. And then you get aspartate reacting with citrulline, and we get arginosuccinate. That's cleaved, and then we get both fumarate and arginine. And in the end, we get urea, and that's what we want because that's what's excreted in the urine. So that's the byproduct of ammonia, and that's what you need to come away with a urea cycle and understand that. If you have a patient who has end-stage liver disease and has hepatic encephalopathy, they used to put them on a branched chain amino acid diet. So branched chain amino acids might be given to patients with hepatic encephalopathy that have high ammonia levels. It's better tolerated than other proteins. They usually treat hepatic encephalopathy with lactulose, which is a sugar alcohol, and it's used for constipation as well. Mandy the Manx was on lactulose until we found an uber fiber food for her that works now. And it binds, what lactulose does is it binds the urea in the colon, and unfortunately these patients often end up with diarrhea. So again, the takeaway is we can't let toxic ammonia levels build up in the system, and that's why we have the urea cycle in liver. If your liver doesn't work, then you're probably going to have problems with ammonia levels. So when we take off an amino group from an amino acid, and we have that alpha keto acid left, we have what we call a carbon skeleton. And that skeleton can be used to produce energy, glucose, ketone bodies, cholesterol, or even fatty acids. Let's talk about hepatic catabolism and uses of aromatic amino acids for a minute. Bear with me. So specifically in the livers where we are now, phenylalanine is converted to tyrosine, and that is an iron-dependent reaction. We'll be talking about the trace minerals. And tyrosine is degraded in the liver as well, and used for synthesis of L-DOPA and other catecholamines. Also can be used for melanin production, that gives us the pigment in our skin, and thyroid hormones as well. Disorders of these two amino acids, one of them you've probably heard of, PKU, fetal ketonuria. If you have a baby, they will test for this before you leave the hospital. The particular individual who has PKU cannot convert phenylalanine to tyrosine. It's common, 1 in 10,000 sadly, and what you end up doing is having to limit protein in general and then add tyrosine to the diet because it can't be converted. Their conversion doesn't occur. Tyrosinemia, you have to restrict both phenylalanine and tyrosine. There's an alkaproteinuria, which is common in Slovakia. I'm actually half Slovak, so this was of interest to me. I hadn't actually heard of this, and the urine turns black when it's exposed to air. Tryptophan is used in the body for a number of purposes: energy, glucose, ketone body production, nicotinamide production, serotonin, melatonin. There is a disorder called alpha-ketoadipic aciduria and glutaric aciduria type 1. So with each of these amino acids, there very often is a specific inborn or metabolic disorder associated with them when they're not metabolized, inability to metabolize them, the body. Sulfur containing amino acids are also catabolized in the liver. We have methionine, which is converted to SAM, S-adenosyl methionine, and that is the body's principal methyl donor. And ultimately, S-adenosyl homocysteine is converted to homocysteine, and then there's a reaction with serine to form cystathionine. There are certain populations, there's a genetic disorder called homocystinuria, and it's common in Ireland, and they have defects in the enzyme cystathionine beta synthase, and they end up with these very high homocysteine concentrations, and this can cause heart disease and promote thrombi formation or blood clots, which is a very bad thing. So ultimately the diet has to be low in methionine, with added cysteine, and supplements of betaine and folate. Propionic acidemia is another disorder found in Greenland and Saudi Arabia, and there's multiple restrictions of amino acids required. So these are quite fascinating, not terribly common, most of them, thank goodness. And I think as our understanding of genetics gets better, we'll be seeing more of these types of disorders being identified. Cysteine is the last one I'll cover. It's used for protein and glutathione synthesis, and ultimately taurine is produced, and taurine is important for our retina and for bile salts, and it's an inhibitory neurotransmitter. And taurine, interestingly, Mandy was just here, and I was talking about how we were using lactulose for her because she had terrible cat story warning, terrible problems with constipation. She actually obstructed. She had a bowel obstruction. She was she was a stray, and she's a Manx, so she doesn't have a tail. And if you think about in a cat's tail, it's vertebra, and there was nerve innervations, no tail, no nerve innervations, we think are probably missing, and she had decreased peristalsis of her bowels, and therefore issues with constipation to the point of having a bowel obstruction. We tried the lactulose, which again can be used to bind ammonia to get rid of it in the body, or for constipation. And we would chase her all over the house. I would, with a syringe of this sticky, sugary fluid, and I still think we're finding puddles of sticky, gooey sugar on the floor throughout the house. Razor blade to scrape it up. So she now is on this very high fiber food, which works magically. And taurine is an essential amino acid for kitties, for felines, and if you're giving them cat crunchies, they have to be cat crunchies. You can't give them dog food because they would be missing this essential amino acid taurine. See how I made it back to taurine in a roundabout way. And I do believe that concludes my lecture. We'll see you next time.